Hello, my name is Deason. My paper is about proteomics analysis of subcutaneous adipocytes. In this study, some abnormalities were observed in human insulin resistance. Adipocytes are cells which are responsible for storing the fat that can be found in connective tissue. So for adipocytes which lacks the ability to take out and store free fatty acid will lead to abnormal lipid accumulation and also abnormalities during the release of adipokines by adipocytes. These are critical factors that lead to insulin resistance and development of type 2 diabetes mellitus. Previous research showed that the mechanisms underlying such abnormalities include impaired adipogenesis, activation of pro-inflammatory pathways, endoplasmic reticulum stress, reduced mitochondrial mass or function, and lipid droplet dysfunction. However, due to the lack of research focus, there are limited understanding on how molecular changes in adipocytes can cause systemic insulin resistance. From recent studies, gene expression analysis of subcutaneous adipose tissue has shown changing patterns of gene expressions. This indicates defects in adipocyte differentiation, cell cycle control, and upregulation of extracellular matrix constituents in subjects with obesity. In addition, downregulation of major metabolic pathways such as branched-chain amino acid, fatty acid, carbohydrate, and mitochondrial energy metabolism, and upregulation of immune response genes can be observed from lean individuals to individuals with obesity and metabolic syndrome. Furthermore, some studies also found that adolescents with obesity were found to have increased ratio of visceral to subcutaneous adipose tissue, which are connected with downregulation of lipogenesis and insulin resistance. This study aims to use unbiased proteomics approach in order to identify molecular factors in adipocytes that could be essential for systemic insulin resistance in insulin-resistant individuals. In addition, this study also aims to identify factors which can distinguish metabolically unhealthy individuals that are insulin resistant from metabolically healthy individuals who are insulin sensitive with similar excess body fat. So the method for this study starts with recruiting individuals with the age from 21 to 70 and body mass index of 20 to 25 kg per meter square, who underwent medical history, physical examination, screening laboratory tests, and 75-gram oral glucose tolerance test. From the test, a total of 23 healthy non-diabetic subjects, which consists of 8 men and 15 women, have completed the study and were instructed to not change their regular diet or exercise for 2 days before the next studies. The subjects arrived to the appointed location after an overnight fast and underwent various processes include body composition measurement by DEXA and adipose tissue biopsy towards the umbilicus through a 1.5 to 2.0 centimeter incision under anesthesia and at 120 minutes euglycemic hyperinsulinemic clamp. After that, all 23 subjects were separated into insulin-resistant group and insulin-sensitive group according to predefined criteria of insulin sensitivity by the euglycemic clamp to prevent any selection bias. Individuals with an average glucose infusion rate less than 5 mg per kilogram per minute during the last 30 minutes of clamp was considered to be IR. In this study, adipocyte proteomics analysis were performed with one-dimensional gel electrophoresis. This is followed by high-performance liquid chromatography electrospray ionization tandem mass spectrometry using a hybrid linear ion trap Fourier transform ion cyclotron resonance mass spectrometer. For pathway analysis, it was performed using Ingenuity pathway analysis, where only the proteins that were identified in more than or equal to 12 of the 23 subjects and differ in abundance greater or equal to 1.5-fold between IR and IS were used in this analysis. A pathway was only considered significantly enriched if the p-value for the pathway was less than 0.01 and the pathway included greater or equal to 5 identified proteins in order to prevent any influence of differences in only 1 or 2 proteins. To perform Western blot analysis, proteins from adipocyte lysates were separated using tris trypsin page before it was transferred to nitrocellulose membranes. The membranes were immunoblocked by GOAT human antibody against fatty acid binding protein, FABP4 and FABP5, with the ratio of 1 to 2500 and 1 to 1500 dilution respectively. The adipocyte size can be measured by mixing well with isolated adipocytes and placed on a siliconized glass slide in silicon well before covered with a glass cover slip. The cell diameter can be measured manually with phase contrast microscopy at 10 times magnification. Approximately 200 to 300 cells were measured for each individual. Based on the predefined IS criteria, the division of subjects resulted in 10 IR subjects and 13 IS subjects, with the average GINF required during the euglycemic clamp differing nearly 2.5-fold. The IR subjects with similar age and gender distribution were significantly more obese compared to the IS subjects. IR subjects had increased level of HbA1c, triglyceride, and non-esterified fatty acid. There is also a trend for increased systolic and diastolic blood pressure and also high-density lipoprotein consistent with metabolic syndrome. The protein were identified in at least 12 out of the 23 subjects. They will differ significantly in abundance with a factor of at least 1.5 between the averages of IR and IS groups. 383 proteins were detected in at least 12 of the 23 subjects. Of these 383 proteins, 94 differ in abundance at least 1.5-fold between IR and IS, of which 30 were statistically significant by unpaired student t-test or Mann-Whitley U-test. The proportion of mitochondrial proteins among the proteins with decreased abundance was significantly greater than that among the proteins with increased abundance. 3 non-mitochondrial proteins with significantly decreased abundance were proteins involved in lipid metabolism, including FABP4, FABP5, and hydrolase domain containing 5, which function in phosphatidic acid biosynthesis and regulates triacylglycerol storage. From the proteomics and ingenuity pathway analysis, there were 8 pathways which were significantly enriched in proteins with outer abundance in IR. Oxidative phosphorylation showed the highest significance, and the manually curated sets of individual exploratory chain complexes suggest 50% to 70% reduced protein abundance of complex 1, 3, and 4 in IR. Complex 3 and complex 4 were considered significant as shown in figure 1b. For complex 2 proteins complex 2 proteins were not detected, while complex 5 proteins were similar between both groups, which is why they are not shown in figure 1b and 1c. The protein set contained adenine nucleotide translocators, and it was approximately 60% less abundant in IR groups. So based on the previous animal studies, the lack of FABP4 and FABP5 protects from IR, whereas increased level of FABP5 in adipose tissue induced IR. As a result, this study also performed immunoblock analysis of FABP4 and FABP5 in 19 subjects from sufficient remaining specimens. Immunoblocks confirm reduced expression of FABP4 and FABP5 in IR by approximately 30% and 50% respectively, as shown in figure 2. Based on previous study, the mitochondrial mass is reduced in white adipocytes or white adipose tissue of IR and high-fat mice. In addition, in white adipose tissue of mice with diabetes, the mitochondrial number, electron transport chain enzymatic activity, oxidative phosphorylation, and beta oxidation were reduced, and mitochondrial morphology was altered. In individuals with obesity and type 2 diabetes compared with individuals who were younger, leaner, and healthier, The mitochondrial number and the expression of key genes related to mitochondrial function were found to be significantly reduced in white adipose tissue. As a result, this study extends these findings by showing significantly decreased abundance of mitochondrial proteins in IR subjects without obesity compared with IS subjects matched for age and gender distribution. From the immunoblot analysis, there was unexpected finding in this study, which was the reduction of FABP5 and to a lesser degree of FABP4 in adipocyte from the IR individuals. Significant differences in FABP5 but not FABP4 persisted in comparisons of IR and IS matched for adiposity. As FABP4 is responsible for regulating transport of NEFA, which increases hydrolytic activity of hormone-sensitive lipase and function as shuttle from cytosol to nucleus for PPAR gamma agonists. The other studies show that mice with deficiency of FABP4 can partially protect against the development of insulin resistance related to genetic or diet-induced obesity. For mice with high level of FABP5 in adipose tissue, it showed a decrease in systemic insulin sensitivity. One possible explanation for the apparent differences between animal data and human data is likely due to species differences. Another possible explanation could be due to the inverse relationship between FABP4 and FABP5 levels, and systemic insulin sensitivity may be mediated through adipose tissue other than abdominal subcutaneous adipose tissue. As a result, this still requires further research in order to understand the relationship between FABPs and insulin sensitivities in humans. Another changes observed in these studies was the increased abundance of several cytoskeletal proteins in the IR subjects. Profilin 1 was the only cytoskeletal protein with lower abundance, which binds to actin and prevents its polymerization at high concentration but enhances it at low concentration. Interaction between the cytoskeletal network and mitochondria play critical roles in mitochondrial fusion and fission, morphology, and localization as well as function. However, this study observed increased microtubule-related proteins but decreased mitochondrial proteins also in insulin-resistant skeletal muscle by proteomics analysis. These observations not only suggest the existence of such reciprocal changes across several tissues, but also possible connection between cytoskeletal and mitochondrial abnormalities in insulin resistance. In conclusion, the new findings found in this study include lower abundance of FABP4 and FABP5, as well as higher abundance of cytoskeletal protein in abdominal subcutaneous adipocytes from IR subjects. In addition, there is finding which confirm reductions in adipocyte mitochondrial proteins. When comparing between adiposity-matched IR and IS individuals, it indicates that the changes in the abundance of proteins are related to lipid metabolism as well as unfolded or misfolded protein response distinguish metabolically unhealthy from metabolically healthy individuals with similar excess body fat. This is my reference for this presentation, and that's about it.